This is Hannah Bauman, Christine Schlaug, and Ashley Lower's final lab project, the effect of dye concentration on a solution's cooling point.
The purpose of this lab is to discover the effect the concentration of dye in a solution has on the solution's cooling rate, if any. This information could be important to certain professions like chefs and bakers, so that they are more aware of how dye concentration may affect the cooling rates of their foods. The independent variable in our lab is the amount of dye 1, blue dye 1, we use in each solution. The dependent variable is the cooling rates of each solution. Our controls are the volume of each solution, the amount of time allowed for each solution to cool, the pressure, the temperature that each solution will be heated to, and the size and type of a container that each solution will be stored in. We hypothesize that if we increase the concentration of blue 1 dye in each solution, then the cooling rate would decrease. This would be because the darker the solution is, the more light energy it absorbs, thus causing the solution to be warmer and to have a slower cooling rate. The dye we used for an experiment was FD&C blue dye 1. This particular dye is both synthetic and soluble. Blue dye 1 is also a part of a group of dyes known as triphenylmethanes, which all have 3 benzyne rings attached to a central carbon atom as their molecular structure.
For this experiment, we used 105 milligrams of FD&C blue dye 1, 700 milliliters of distilled water, one 100 milliliter graduated cylinder, 7 150 milliliter beakers, 1 large heating plate, 1 scupula, 1 glass stir rod, 1 Celsius thermometer, 1 scale, 1 pair of hot hands, 1 stopwatch, 3 pencils, and the necessary collected and tabulated data tables. Prior to doing the lab, we dissolved and diluted the blue dye 1 solutions to the necessary concentrations.
For our procedure, we heated each solution to 50 degrees Celsius using heating plates. Then we removed them from the heating plates and allowed them to cool for 10 minutes, and we recorded the final temperatures.
We collected our data in a table titled Temperatures and Observations of Solutions. Each solution was heated to an initial temperature of 50 degrees Celsius, and during the heating process of each solution, steam and condensation were present. While the final temperatures for each solution differed slightly, they were all relatively close to 40 degrees Celsius.
We processed our data in a tabulated data table titled Temperature Changes and Cooling Rates of Solutions. Here we calculated the changes in temperature and the cooling rates of each solution.
The line indicates there's a slight increase in the change in temperature as the concentration of blue dye increases. However, as shown by the plots on the graph, there is no apparent pattern to the effect of the concentration on the temperature change.
From our results, we've concluded that the concentration of a blue dye solution has no effect on its cooling rate. There was also no noticeable pattern in our collected data. We initially hypothesized that if we increase a solution's concentration of blue dye, then its cooling rate would decrease. Our data and results disagree with our hypothesis because there was no apparent pattern to our data. This could possibly be due to errors we had accidentally made in our procedure. One random error we observed was the inaccuracy of the Celsius thermometers. After allowing the solutions to cool, the reading on the thermometer was still not clearly determined. This would have affected our changes in temperature recorded. To fix this problem, we could obtain better and more precise thermometers. A systematic error that occurred was having to allow the solutions to cool after overheating the required 50 degrees Celsius. This is an issue that could have been controlled by following the procedure more closely and carefully. It's important to know that the concentration of a dye in a solution does not affect the solution's cooling rate because now certain professions that handle dyed food, such as chefs and bakers, can rest assured that the concentration of dye in their products will not affect those products' cooling rates.